topic for today is nitrogen and protein determination with accuracy and factors affecting the accuracy. This slide shows different outlines of this session. We will discuss one by one during this session. First of all, objective of this session, we will talk about accuracy, precision, different determination steps, different factors that may affect during nitrogen determination, and we will talk about systematic and random error at the end of this presentation. First of all, accuracy. Accuracy is the closeness of a measured value to the true value. If you get control recovery near to 100, so that means you are hitting the target true value and you are getting the actual value. Precision Precision is the agreement among the replicate of same sample, and RPD and RSD determine the precision. In this slide, there are two examples. In first example, a sample having actual concentration 18.66% nitrogen is analyzed in replicate and observed values are 18.6, 18.64, 18.66, and uh, so for this data, recovery is 99.85% and RSD is 0.16. That means this data is very accurate and precise, and that means you are getting actual value. In second example, a control sample having actual concentration 35.6% protein is analyzed and triplicate, and results are 26.4, 26.8, 26.6. In this case, RSD is 0.75, but recovery is very less, and that is 74.7%. So in this case, this data is precise but not accurate because this data is very far away from actual value or from target point. So a given set of analytical data can be said of high quality when these data are more accurate and more precise. That means the measured replicates should be more close to true value and more close to each other. In this case, this data can be called as of high quality. This slide shows different steps of determination of nitrogen. First step is sample preparation. So grind the sample and homogenize the sample. Take nearly 1 gram of sample, transfer the sample to the digestion tube, add 1 tablet of catalyst, add 10 ml of concentrated sulfuric acid and put these tubes on the digester as shown in this figure. So next is digestion. So do the digestion for 60 to 90 minutes at 420 Celsius temperature. After digestion, next step is distillation. So in this figure, distillation apparatus is shown. In distillation apparatus, you can see two areas. The receiving area for this receiving flask, you can see this green color, and second portion is for this distillation tube. So you will put distillation tube here and you will put receiving flask in this receiving area. So uh, in this distillation apparatus you have in the screen you can see different programs. So these programs are already set, or you can save a new program according to your requirement. Set the distillation time 5 to 6 minutes, steam 100%, alkali volume 50 ml, dilution water volume 80 ml. So then put the put your receiving flask, put your digestion tube and on this distillation apparatus, and it will take 5 to 6 minutes, and you after 5 minutes you will get distillate in green color. So here I will discuss how to prepare receiving flask. So for preparing receiving flask you need 3 to 4 solutions. First solution, you need boric acid solution 4% or 1 to 4%, and then 0.1% bromocresol green indicator you need, and you need 0.1% methyl red indicator. And from bromocresol green and methyl red you will prepare mix indicator in the ratio of 10 ml of bromocresol green and 7 ml of methyl red indicator. So from this mixed indicator you will take 10 ml of mixed indicator and add this 10 ml to the 1 liter of boric acid solution. Then from this boric acid solution take 50 ml and put into the receiving flask. So then you will on this distillation machine and you will get after 5 minutes you will get distillate. And then next step is titration. Titrate this distillate against standard acid, normally 0.2 normal HCl or sulfuric acid is used. So this is all about this digestion and distillation titration process. If you have sample of salts like ammonium sulfate or any other salt having ammonical nitrogen, so for this salt no need digestion, so do direct distillation and titration. This slide shows different component of AQC, analytical quality control. AQC is the set of measures or set of activities that ensure the quality of data. So this slide shows component of AQC. So AQC component need normally for all laboratories. So here I have discussed in general, but next slide will show this slide shows the factors affecting the accuracy of nitrogen and protein determination. First one, competency of lab personnel, method selection, validation, high purity chemical and reagents, calibration of analytical weighing scale. Then standard solution, digestion, distillation. So these are the factors that may affect the determination, accurate accurate determination of nitrogen protein. So if you have not standardized your acid, and even with the difference of 0.05 there would be difference in nitrogen and protein concentration as shown in this slide. There will be difference of 0.5% in nitrogen and 2.5% in protein. Distillation, if you do this next digestion, if you do digestion for more time, so it will also lessen the nitrogen content, and it will affect of course on accuracy. Distillation, so for distillation, for proper distillation, at least run one empty tube or blank sample in the very start of distillation process. Otherwise you will get less distillate. If you will get less distillate, even you are giving 5 minutes for distillation, but you in very first sample you will get very less distillate, and in this slide you can see on the right flask there is less distillate. So in this case your accuracy will be affected. This slide shows different types of errors: systematic error and random error. Systematic error arises from imperfection of instrument or mistake of analyst. Normally, analyst can make mistake during titration or during the identification of endpoint. So that will be systematic error as shown in this slide. On the right side, there is over titration. Then next is random error. Random error arises due to the limitation of equipment. Limitation of equipment, as I have shown in this slide, on the right side you have less distillate in the very first sample. So if you have not run the blank or you have not run the empty flask and you run you have run first sample direct, so you will be having less distillate and you will be having less concentration, you will be having less recovery. So this is actually limitation of this machine in the very first sample. Why? Because in the very beginning this distillation machine need some time to raise the temperature for steaming. So normally it take time 1 to 2 minutes. That's why it affect on the distillate. So this is random error. In this slide you can see some guidelines, or you can see some point that you can consider. So consider the factors that are discussed earlier. Follow the procedure exactly. Remove the systematic and random error as you can. Run the internal quality control samples that will assure the accuracy and precision. Normally blank is used to remove reagent interferences. Control is used to check accuracy of the analysis performed. Spike is used to check the matrix effects. Duplicates are uh, run to check the precision and repeatability. So I am very thankful to you for, to watch this video.